you, everyone. I would like to start first thanking for the opportunity to be here. It's actually my first time in the ISMB and being part of this NetBio. So really happy. And just a little bit of myself. So I've been working with single cell for about 3 years now, and mainly I've been doing a lot of collaboration, analyzing data from different things like the differentiation rare cells in the midgut of Drosophila, RNA velocity of organoids, and many things. And because of that kind of experience, I noticed that the network of genes is actually not much explored. And then I found this, okay, so why not dig into this part? So from 1 year and a half, I've been working on the communities of the detection, community detections, right? See the mouse? Sorry. Let's see the. Ah, okay, okay, thank you. Yes. So I start with the question. So having a graph, so a network. So how do we cluster this, right? So we first start identifying the nodes. So the nodes in this case are um, co-authors, and then the links are the co-authorship in papers. And then, as you can see, it forms some communities, basically from the departments. So, which is like they more likely to collaborate to each other than to other departments. And fast forward to nowadays, we have a single cell, which is very powerful, provides a lot of resolution for the identification of gene expression. And indeed, we have cells and by genes matrix, and then from there we can reduce the dimension and then try to cluster cells. So these are the main approaches in most of the experiments. But we can also focus on the other dimension, that we can focus on the genes and then also try to cluster networks of genes. So this is the point that we are going to work in this project. So how to cluster gene? Uh, in this case, there are many methods, but we are going to use modularity. So modularity basically measure how good is a structure, a community structure in a given network. And then, for example, when we look to here, if we cluster this network here in this way, we can see that, okay, this is not a good one. So, but this one is a better one. So by having this modularity, we can kind of understand how good is a cluster. And then when we have from the network, we can create the adjacency matrix, and then we can calculate the modularity like this. And then, so the problem now is how can we maximize the modularity, since we want higher score to find better communities, right? And but the problem with a single cell or larger data sets is that sometimes the community is too large, or the network is too large. We don't have computational power, but then thanks to new algorithms like Louvain, Leiden, we can have much more power and also computationally. Um, but then with single cell also brings the problem with the sparsity, dropouts, and the low correlation, since we don't have much signals. So there are another layers of difficulties that we cannot apply like the previous methods used in microarray or bulk RNA-seq. So there are new is a need for new methods. And some of the new methods actually focus on a very few genes, like 500 genes, or top highly expressed genes, like 2000, 3000 at most. And well, but what about the other more than 20,000 genes, right? So we don't look to them. So it's necessary to develop a method that can extract functional modules from sign networks using single cell RNA-seq. And sorry, here sign network is because we have the positive correlation uh, between genes, but we also have the negative correlation, and especially in single cell, where we kind of, for example, the marker genes, right? We know when even in a given sample, if we know that this marker gene is expressed in this subcell type, but not in that one. So this can be used in favor for our identification of communities, but it's not being much explored. So this definitely can help. And so then I would like to introduce our tool called the low vein sign. Basically, we have expression matrix count from single cell, and then we filter out the genes that are basically zeros. So we have a bit smaller one, and then we calculate the gene scores. We in the lab we have developed this tool EISP, which generates codependency and exclusive expressed index, which mainly binarize. So when we have observation of expression, we assume CDI, like codependence. And then when there's the mutual exclusivity, we it's infer the EI, which is the negative correlation. And then from there, we have basically two networks of positive and the negative ones. And then we use a little bit of filter on that to reduce the numbers. And then finally, we have our low vein sign that adapt the positive and the negative, and then also consider some other parameters that I will explain next. And then we have the partitions, and then these partitions we can find out how many communities, how genes, which genes are included in which communities, and then so on. So we can do downstream analysis. And yes, so let me explain details how we find out about this low vein sign. So as I explained, modularity can be identified like this one, and was proposed by Newman Girvan, and it's been used, since then. But then later on, Gomez introduced. Okay, so how about when we have the sign networks? If we have a positive one, negative one, we just subtract um, the negative one, and then we can have a better identification of communities based on this new modularity. And yes, it works in networks that are based that have a similar amount of negative and positive ones. So it's mainly maybe social networks. But when we have more unbalanced networks of like negative ones, it doesn't work well. So then Trag also tackle this question and then try to improve how can we identify communities, then including resolution, in this case the gamma here. So by modifying the resolution in both positive and negative ones, we can find larger or smaller communities. So I think if you have worked with Surat or Scampi, and then you may be familiar with the resolution to find more or less clusters. So it's basically the same idea. And then, yeah, this also works in a way that we can move this, but it's also not very optimal for the amount of negative networks. And then in 2013, so Sugihara proposed this method, which is it's a way to balance how much we can put strength on the negative ones or the positive ones together. And then this came out, okay, so what about we don't put everything together? So we just integrated this alpha balancing, but also includes the resolution so that we can adjust, since uh, single cell data is very variable. So it depends on the kind of data you work with. It has a lot of complexity. It makes different network than a much simple data set. So we thought, okay, let's try this method. And we call it Lovine because we use Lovine for the modularity um, calculation. Yes, to as a very fast approach. So I don't think I have to explain about the Lovine method. And okay, so then the results I will show here, I'll just explain a little bit how is the Lovine and Lovine sign, the difference. So Lovine, basically we focus only in the positive links like this one. So when we look to gene expression data like this, the yellow is the expressed. We can form some connections, and then from there we have a network, and then this network we look for the modularity and then communities, right? But our proposal, we include the negative ones like this one. So how this will differ in the results in this example? Mainly, Louvain will find two clusters like this one. But in ours, since we can balance the positive and the negative ones, we can find actually two or three, like small clusters here. And then actually, because of if you put more weight on the negative one, these will shrink this bigger cluster into a smaller one. So why is this important? So if you worked with functional analysis, you know that if you put 1,000 genes into geo enrichment, it's not very meaningful, or it's actually very vague. So having large clusters of communities of genes, it's not very useful to identify or later on. So having a method that we can reduce the communities is also easier for the interpretation. So this method also helps in this way. So to prove that this works or not, we tried the simulation validation. So we created this very simple community network formed by 4 communities, and then we removed the labels, and then we ran several popular community algorithms here, and then our is the second one, found like a very high with confidence the correct labels in a very fast time. So we found like, okay, this maybe too good. I don't know. I'm from the biological background, so I think still things that we need to be considered here. So if you know, if you have some ideas, please uh, let me know. But anyways, for this kind of problem, it solved well. But then, okay, so how about we apply to real data sets? Then we apply to popular data sets that are assigned. So I'm not sure if you know, if you're, but they are quite popular. And the opinion sign social networks, with they have trust and distrust, and slash dot when they have like they classify into friends or enemies. And then the Wikipedia, which has the positive and the negative votes. And then all of them, sorry, it's a bit small here, but if you look to the red and the blue one will show the positive connections in the community. And red one will show, sorry, it's opposite. So the red one will show the intra communities, in, inter communities connections, and the blue one intra communities. So what does it mean? It's like the more towards the right side, it will reduce alpha, it means it will put more value on the on the negative one. And then basically we found that the contribution of alpha can be helpful to identify better communities. So I move forward because of time. And then we also applied to the PBMC data from the CIRAT. So it's very popular. And then where we isolated the CD4 subtypes, where we have at least 3 types there, right? So if we use this data set, can we find better communities, or can we find more specific communities? And we found that the Louvain sign that we create can actually find smaller communities, but not just smaller communities, but it also isolate those that are mutually exclusive or marker genes better. And this is just an example of what is mutually exclusive genes. And the last example is using a more challenging data set, which is a glioblastoma, and that is a time series data. And yes, in this data, we also could observe how the modularity changed towards the parameters we have, but also the time series. And we found a very, very conserved clusters of genes in this example here. So if you look to the Sankey plot, most of the clusters are conserved through the time. And then also the geoterms identified here are also very, very close to the function of the cells. So yes, and our approach is basically data-driven. So we don't use any particular database to build on top. So I think that's a very helpful. And I didn't include here, but we also find the database we use is the marker genes for cell atlas, cell markers 2.0. There we can find enrichment of clusters with more specificity to the disease in this case. Yes, so today I stop here. So these are our take-home message that the Louvain sign can include more sensibility than the original Louvain. And when we use the marker genes, they're enriched in some specific functions that may be more related to the disease or to biological functions. But it's still some limitations that we are trying to tackle, like how to find the optimal parameter threshold. It's a very difficult question. And then also the subset of the counts, also a difficult topic. So yes, I'd like to thank everyone, or the organizers, for the opportunity again, and also my laboratory being so supportive. Thank you so much.
So we do have time for 1 or 2 short questions. Mehmet, you want to start?
So how were the simulation studies conducted? So what is the generator model for the simulated gene expression matrices? Or the simulated networks, right? So you are directly simulating the networks, but there is an underlying gene regulatory network. So do you use a gene regulatory network and simulate gene expression based on it to construct a co-expression network? Or is the co-expression network simulated directly?
No, we simulate directly. It's actually not it's a very simple this one, right? Yes. So it's we created this synthetic validation. We basically have we define like, okay, so we want 4 clusters and we want to have this percentage of positive and negative ones, and then we create that. So it's very simple. It's very simple.
So essentially, they are supposed to be, the pairs are supposed to be either positively correlated or negatively correlated.
Yes.
So a related question is, in a pathway, right, some genes will be negatively correlated, strongly negatively correlated. Some genes will be strongly positively correlated.
Right.
So an alternate approach to all these is just taking the absolute value, because negative correlation also means functional association. So did you check whether there is any advantage to incorporating the signs versus just looking at the magnitude of correlation?
Yeah, good question. We haven't, yeah, checked this, but
Thank
thank
you.
you.
I wanted to ask if you did any transformation of the correlations that you use to build the gene networks, and especially which cutoff did you use to determine a link between two genes? Like, did you use any number greater than 0 for the positive correlation and any
Right.
number below 0 for
Yes, basically yes, but we filter out those are too many zeros.
No, I mean when you calculate the correlations between the two genes, like, do you have a minimum correlation?
Okay. Yes. So this approach here, it will calculate. It's a log-based rank score, and then we basically use 20% of that contain like 80% of the edges of the network. So then we can reduce and don't rely on a specific number. Yes.
Thanks.
Thank you.
Well, thank you.